generation scientists. And in the previous sessions, you might have understood about how pharmacological tools that may be helpful for the drug discovery as well as the development. And herein we are trying to explore how computer-aided drug discovery will be useful in the design of novel drugs as well as how they are helpful in the drug development process. And typically, there are certain things which we will be exploring today. Me, along with Dr. S. Dilip Kumar, who is the associate professor here, we will be exploring these concepts. And before entering into our major topic, homology modeling, I just want to give some basic idea about what is computer-aided drug design, why it is essential to have computer-aided drug design, what is the need for computer-aided drug design, and how specifically we will conduct a homology modeling, followed by Dr. S. Dilip will take some examples and explore the workshop. And drug discovery is a challenging process because there are many steps that are involved in the drug discovery. If you see, traditionally, the drugs were not discovered rationally, we can call. Say, for example, previously the drugs were developed based on certain therapeutic activities. Say, as all of you know that in the olden days, most of the drugs or most of the plants are some sort of extracts of the plants are taken and they are used for the treatment of certain diseases. So that is called as folklore use of natural products. The natural products are replaced by different, scientists or doctors at that time or the physicians. They just use a certain plant parts and based on the observations, they will say that this is used for so and so. Some type of discovery has led to the discovery of one of the plant known as distalis, which has glycosides that is used for the treatment of congestive heart failure. Not only that, there is also serendipity. There are many drugs that have been developed by serendipity. One of the breakthrough observation that has been seen as a well-known example that Alexander Fleming, who has observed that the fungal strains have released certain antibiotics which will be used to kill the microbes. Based on the serendipity also, the drugs have been developed. And later, after some rise in understanding, the scientists used different plant materials or the natural products, and they screen a large variety of the compounds. And after screening a large number of the compounds, they may found one lead molecule that maybe has the ability to convert into a drug molecule. So, like this, there are these are some of the ways in which the drugs have been or the lead compounds have been discovered. And if you take the typical drug discovery process, it is time-taking as well as a costly affair. It approximately, say, you will start with 10,000 compounds and you will filter after certain preclinical phases, filter them up to 250 compounds. After that, whenever you get that 250 compounds after entering into the clinical phase 1 to 4, we will get only 5 compounds. From that, ultimately, we will get one drug molecule. So, the whole story here is in order to find one drug molecule, it requires a lot of effort in terms of energy, time, as well as money. And the number of compounds you are screening here, 10,000, it's not an easy thing. Because in order to screen 10,000 compounds in experimental animals, it requires a lot of money and effort. And ultimately, you are able to develop only one drug, which will be approved by the FDA. And the estimate is that the drug market, the drug that will reach in the market are 1 in 10,000, and the cost is around 300 to 500 million US dollars. And time required is more than 10 years, it will approximate. And there are also chances that after reaching into the market also, we have post-marketing surveillance where certain drugs will show toxicity and we will take back the drug from the market. So these are the things which are creating some negativity on the drug discovery. That is why we need a more rational approach where we can cut down the number of compounds, we can cut down the cost and make it effective in terms of cost, time, as well as effectiveness of the drug molecule without any toxicity. So for this, we have computer-aided drug design. So if you see the significance of the computer-aided drug design, computers, as I said, they are playing a crucial role in drug discovery, and they are helpful for identification of cost-effective drugs. That means with less cost, we are able to develop, top class of the drugs. And in silico or the computational methods are reducing the use of animal models. I told that in order to screen 10,000 compounds, you require a large number of the animals. But computers will help us to understand which is a promising drug-like molecule. As computer is already screening that, we will not go for animal testing. That means it is cutting down the animal testing. That means it is cutting down the impact on the animals also, so that we can also save many animals. That is another thing. And not only that, it is also useful for rational design of novel and safe drug candidates. And it is also useful for reposition of marketed drugs. That means there are certain drug molecules which have shown promising activity, say for one type of activity. Say one drug molecule is used for the treatment of cardiovascular drugs. And the same cardiovascular drug, it is well proved, it is very well adapted to our body. You can try that molecule against coronavirus. That is called as repositioning or repurposing of the molecule. That is, we are repurposing or seeing that molecule if it has a different purpose. And it will support extensively the medicinal chemist in the drug discovery process, because it is having very great advantages. And the other advantage is not only decreasing the pharmacological screening, it will also minimizes the synthetic part. The number of compounds you synthesize will decrease, and the biological testing will decrease. And we will concentrate more on the most promising compounds. Most promising in the sense, the compounds or the lead molecules which have the best activity with low toxicity. This can be predicted by using the computational. That is the advantage of the computation. I can simply say it's a dry method. Dry in the sense, we are not going any thing into the lab. We are not synthesizing a compound. We are not testing it in the animals or some other cell lines. We are doing everything here within the computer. That means I can call computer-aided drug design. It's a simple dry method. In the dry method, you are able to cut down many things. Whenever you are able to cut down many things, the number of things which you operate, the time required for the operation will be decreased in the real-time experimentation in the wet lab, right? Not only that, besides explaining the molecular basis of the therapeutic activity, it also predicts possible derivatives that will improve activity. So if you take a lead molecule, the lead molecule can be modified in the sense within the computer will change different type of substance. Whenever we change different type of substance, we'll get a different derivatives, and the derivatives we can also predict the activity and toxicity of the derivatives, right? That is why we will explain the molecular basis of the activity, as well as it is also possible to predict the derivatives which are having the best activity. And we have different techniques like the molecular docking, homology modeling, the three-dimensional quantitative SAR, and the molecular dynamic simulations, and ADMET predictions. And all these are the different types of in silico tools. In silico means simply in box. Everything is done in the box. In the box in the sense, in the computer. That is as simple as that, right? Techniques, these are the extensively used techniques in the drug discovery, right? So what are the advantages of drug discovery? As discussed earlier, for experimental testing, we need smaller set of compounds from a larger compound libraries. Previously, I told that for testing the compounds, you need large number of the compounds. But the computer is able to discover which type of compounds are better. That means you will filter most of the compounds. The computational methods are able to filter majority of the compounds, and we are only using little number or smaller number of the compounds that may be having the potential activity. Then we'll go for anyhow we'll go for the experimental testing in the laboratory, right? And we can also predict the drug metabolism and pharmacokinetic properties like absorption, distribution, metabolism, excretion. And we will also assess if there is any chances of the toxicity. And we can also optimize the lead compounds. So these are the advantages. We are using smaller set of compounds. We are able to predict the pharmacokinetic and toxicity profile of these small set of the compounds. And if there are any toxic functionalities in this small set of compounds, we can also optimize the lead molecule. So these are the different advantages of computer-aided drug design. And here the designing of the novel compounds can be achieved either by growing starting molecules one functional group at a time or by piecing together fragments into novel chemotypes. What we can do is we can select certain important pharmacophores or the privileged structures which have better activity. We can grow on that molecule, or we can compare two privileged structures, and we can combine both these two, and we will get a novel chemical type of compounds, and that molecule may have a better activity. That can also be screened by the computer-aided drug design. And the traditional experimentation, as I said, it requires animal and human models can be replaced by computer-aided drug design, saving both time as well as cost, right? And one of the major problems with majority of the drugs, particularly the drugs that are used against cancers, as well as the microbial infections, is that drug resistance. Because if you take cancer and antimicrobial resistance in the sense, there are many antibiotics that are available in the market. And say within India, the scenario is that you can go to the shop and you can ask for any antibiotic. They will just give like. So that there is no rational in using of antibiotics. Whenever somebody is feeling some cold, what they will do is they will go and take certain antibiotics from the medical shop, just by saying I have so and so symptoms. So it is not prescribed, right? In such a cases, whenever you irrationally use the drugs, there is a greater chance of drug resistance. And we can land into we are close to the pre-antibiotic era. Before the development of the antibiotics, there has been an extensive cases which have been death rate because of just a formal infections also. Then after discovery of the penicillins and the other antibiotics, the scenario has changed. But now we are irrationally using the antibiotics. Again, it is causing this drug resistance. Similarly, the cancer, one of the second leading cause of deaths worldwide, it is also having, majority of the drugs have the drug resistance, right? And we can also see that we can design compounds in such a way that they are having less drug resistance, and they will be an important lead molecules that would target the exact factor that is responsible, because we are going deep into the protein with which the drug molecule can interact. The CAD also leads to the construction of high quality data sets and libraries that can be optimized for high molecular diversity or similarity. And this will be helping the construction of a variety of chemotypes or the data sets and library of the compounds that can be optimized for differences as well as the similarities between that particular chemotypes. So basically, if you see the lead identification by computer-aided drug design is done by two methods, that is ligand-based virtual screening and the structure-based virtual screening. The structure-based virtual screening, in structure-based virtual screening, we have the knowledge of the structure of the protein to which the lead molecule can interact. So if you take a disease, there are different types of proteins or the biomolecules that are whose alteration is responsible for the disease. So we know that if we inhibit that particular enzyme, we will get a potential lead compound. Okay, and for that we know the knowledge. We have a knowledge of the structure of the protein. That is why it is called as the structure-based virtual screening. Again, the virtual means what? It's not real. It's not real means it is not. We are not having any screening in the laboratory. and all this is dry screening. That is, we are doing everything in the computer, right? So here we have the knowledge of the, structure or the protein or the receptor, and we will identify the binding pocket and dock the molecule at the active site where the catalytic process. Say, for example, if you take carbonic anhydrase enzyme, and similarly, if you take beta lactamases, right? All these enzymes have an active site. The active site will be inhibited because we have a better understanding of this particular structure of that protein, and we will design based on the groups that are present, because proteins are polyfunctional, which contain different amino acids. We know the active binding site or the pocket, and in the we will try to see the look into the pocket sites chemistry. Based upon the chemistry of the pocket site or the active site, we will design our molecule and see and dock this molecule into this and see if it is fitting properly and it is having the ability to inhibit or not. Based on that, we will prepare a series of the derivatives and see which compound is better. And after that, we'll go for toxicity predictions. If it found to be safe computationally, and if it is having better binding activity, you can synthesize it in the laboratory, and then you can go for the wet methods of the screening, nor after this virtual or the dry methods of the screening. So here we have a knowledge of the protein, but in ligand-based drug design or the virtual screening, we don't have the knowledge of the protein. Here, ligand-based drug design explains the strategies wherein the information from existing ligands of a drug target is utilized. Say, for example, if you take COX enzyme inhibitors, all the COX enzyme inhibitors contain a carboxylic acid. That is the common structural feature. So we have a certain group of molecules where they have a common structural features of the ligands. That particular common structural features of the ligands will be taken into consideration, and based upon that, we will create an ligand, the best ligand, based upon the drug target that is utilized. And after ligand-based virtual screening, where we have got a series of lead compounds as well as structure-based screening, we'll go for prioritization, right? In prioritization, what we do is we should we can't take all the molecules that are high. We should assess for its drug-like properties, toxicities, as well as whether it has a good pharmacokinetic profiles or not. And we will filter the compounds, and after filtering the compounds, we'll get some molecules. That particular molecules are actually entered into synthesis as well as experimentalism. So these are the two basic things we have in we utilize for the identification of lead compounds by compute-rated tools or the compute-rated tools. And if you observe, there is a lot of success of this computer-aided drug design. That is why it is more used nowadays, and it will be utilized in the future also. 12 small molecules like ACE inhibitor, captopril, as well as protease inhibitor like saquinavir, and anti-flu agents like janamavir, oseltamivir. And these are some of the drugs that have been approved for therapeutic use, and they have all been developed by using the computer-aided design strategies. So you can see the impact of computer-aided drug design. So it itself says that we can depend on computer-aided drug design because it has enabled, this technique has enabled to deliver certain drug molecules into the market by saving a lot of money as well as the time, as well as human effort, and we are also not harming many animals, right? This is the advantage of the key strength of the computer-aided device. And this is another molecule that is known as dorzolamide. The dorzolamide is a type of agent that is used for the treatment of glaucoma, which is a carbonic anhydrase inhibitor. And not only that, there are many other drugs which have been developed by utilizing the structure-based drug design. There are also certain drugs that have been developed with the help of ligand-based drug design. Say, for example, the nalidixic acid, norfloxacin, as well as losartan, the angiotensin 2 receptor antagonist. All these are the drugs that have been developed based upon the ligand-based, by utilizing the ligand-based drug discovery. So it is crystal clear that we can use computer-aided drug design for developing new drugs. But there are many things which you need to understand in order to design and develop new drugs, for which this workshop is. And the main aim and objective of the present module is, and this is introductory in scope. We are not going into the details, but this introduction is very essential for the next generation researchers who are much interested in utilizing these tools for designing and developing a new type of molecules for the treatment of variety of diseases. Because we have many challenges, right? There are many challenges where we need to develop a large number of the drug molecules. There are cases where patients are dying because of even small things, small disorders. There are certain rare diseases, there are certain tropical diseases, and there are also complex diseases like cancer and the cardiovascular disease. So always the research window is open where the medicinal chemist or the scientist can work and develop new type of drugs that will be helpful for the humanity, right? So that's why we should have a good understanding of this computer-aided drug design. So it is introductory in scope, and it is aimed at beginners in the field of computer-aided drug discovery. And the objective of the workshop is to familiarize the participants with introductory concepts of CAD so that they are equipped to learn advanced techniques on their own for application to more complex systems or complex problems. And the expected outcome of this module or the workshop is the participants should be able to perform homology modeling and molecular docking simulation studies on different targets of the universe. So today we will discuss about homology modeling. I'll just give a brief idea about what is homology modeling, followed by the actual, by taking one example, how we can operate this different softwares that are available in order to conduct or perform this homology modeling. So homology modeling is a condition where, say, you know a target, but you don't have a crystal structure of a target. Just now I said that in structure-based drug design, we have a structure. If you have a crystal structure of this molecule already in the protein data bank, we in such cases, we won't go for homology modeling. You know a target where the potential lead molecule can interact, but you don't have a crystal structure of a protein. Because it's not easy to get a protein all the time, the crystal structure of the protein. Because why? Because the protein is not a separate identity which is present in the cell. It is the protein itself is a polyfunctional thing which has many functional groups because of large number of the amino acids. And due to that large number of the amino acids, it have different functionalities. As it has different functionalities, it is associated with different proteins or other biomolecules within the physiological system or the cell. As it is associated with that, the isolation of the protein into a purest form is one of the problem. And other important problem is, even if you isolate that protein in the purest form, the crystallization of that particular protein is really a big problem. In such a cases, what we do is we use the computational tools and we will conduct the homology modeling. So if you take the schematic outline of the application of homology modeling and X-ray crystallography in this drug discovery process, say this is a target protein sequence. Whenever you have a crystal structure of the target protein, which have been already determined by X-ray crystallography, then what you will do is you will start screening the compounds, virtual library of the compounds, and you will perform the molecular docking. After performing the molecular docking, you can confirm using the crystallographic as well as the kinetic analysis the best compounds, right? After identifying the best compounds through the kinetic analysis, we'll start identifying the lead molecule, and we'll also simultaneously optimize. After lead identification, we need to optimize that. If it has better drug-like, that is what we have to, I have told that prioritization, where we are trying to what? Optimize the lead molecule, where we will only take the best candidates in terms of both pharmacokinetics, pharmacodynamics, as well as the toxicity aspect. After this, we'll go for compound development, actual drug molecule. And if we found that we have identified a lead molecule and optimized the lead molecule, and we found that it has a good hit molecule, we will start synthesizing that particular molecule and its derivatives in the laboratory and do the actual screening in the laboratory. And this is a normal process whenever you have a crystal structure of the target protein. But it is not the case, because I told that there are limitations in getting this crystal structure of target protein. In such a cases, we will rely upon the homology modeling of the target protein. Right? Homology modeling is one of the computational structure prediction. Here we are using computational tools to predict the protein structure that are used to determine the 3D structure of protein from its amino acid sequence. So why homology modeling means the rate of structure solving through NMR and X-ray is slow compared to the deposition of DNA and protein sequences. If you want to determine the structure, or if you want to, resolve or determine the structure of a protein by using NMR and X-ray spectrography, X-ray crystallographic techniques, it requires a lot of time. But homology modeling will save that time. And the membrane proteins are difficult to crystallize. As I said previously, it is not an easy case. As the proteins are polyfunctional, they are associated with other proteins as well as other biomolecules. It is difficult to crystallize. And in particular, if the protein of your interest is associated with the membranes, like the G protein coupled receptors, it is even difficult, because G protein coupled receptors are the large family of the receptors which are widely distributed throughout our physiological system. So this is one of the hard target for the development of drugs against variety of disorders. One of the limitations is, as it is associated with the lipid membranes, the isolation of that particular G protein coupled receptors is not an easy task. Even after isolation, crystallizing it into a form where we can use the X-ray crystallographic technique for its structural determination is really a tough job, right? And for understanding the activity of a lead compound, the knowledge of the 3D structure is essential. 3D structure of the protein is essential. So based on the 3D structure only, the function of the protein is dependent, so that we can see whether our lead molecule or the molecule under consideration can alter its protein function. And structural information further enhances our understanding of how this protein is interacted with other protein, or whether it is interacted with other DNA molecules, right? So all these will help us to design better drugs and deliver the best molecules in all the terms. And coming to the basic steps involved, the first and foremost step is the template identification, followed by the alignment of the query protein with that of the template molecule, template protein, and we'll generate the model based upon this. After generating the model, we need to optimize whether the developed model is a better one or not, and we should also assess or validate if it is suitable model in order to perform our molecular studies. So coming to the first and foremost step, that is template protein identification. Template protein identification, it is done by local alignment. We have two important softwares like BLAST, that is a basic local alignment search tool, as well as the FASTA. Both these programs can be used to search the proper template in the protein database for query protein sequence. So here you need to understand two things. One is query protein and the other one is template protein. Template protein is a protein whose crystal structure is known. Okay, query protein is a protein for which we are trying to do the homology model. So here template protein and query protein are more closely related. They are not they are different, but they are closely related. But here we have template protein which is already isolated, crystallized, and its structure has been well defined, and it is stored in the protein database. Now we are interested in some protein which is related to the template protein, which is called as a query protein. We are trying to sequence this protein, and we are trying to model this protein. That is what we are doing. So we need to understand what is query protein. We need to prepare a model. That is what homology modeling. Homologous means, that is homo means same, logos means resembling the sort of template protein. They are not exactly the same, but they are closely associated, right? So the best template structure, the one with the highest sequence similarity to the target, will serve as the reference of the template. The one which has close association or structural resemblance will be the template for our template. First step is we need to identify the template protein, which is located in the protein data bank, which is having correlation with that of the query protein. Once we have identified a template protein, then we need to see the sequence alignment. That is, we try to match the template protein with that of what we say, our query protein. The sequence alignment of template protein with query protein can be carried out by using Clutsolve. This is another software. The generated alignment can be manually adjusted in order to align the regions which are conserved or transmembrane regions in case of the G protein coupled receptors. Conserved means, say for example, I'll go here. Yes. If you see this, this is the actual, template protein, and this is our query protein. If you see, whenever we are arranging the query protein, there are certain, it will be conserved, there are certain gaps. That particular certain gaps that have been identified here. Even if you see these, there are certain gaps here, right? That particular gaps, just a moment. That particular conserved or the transmembrane regions can be manually adjusted. That is what we can do using these softwares, right? In order to align the regions which are conserved or transmembrane regions in the case of the G protein coupled receptors. After aligning this or matching the sequence, the next step is modal generation. That is nothing but the actual framework construction. If you see that here you can see the yellow colored protein as well as the protein with different colors. The one which is in yellow color is our query protein, and the one the other with different colors is the template protein. We have identified the sequence alignment, and now we are trying to generate this model based upon the matching. The construction of the framework is computed by averaging the position of each atom in the target sequence based on the location of corresponding atoms in the template protein. We'll try to match both the template protein, we'll try to match the query protein with that of the template protein. And we'll try to position all the atoms whether they are perfectly aligned or not. Whenever they are perfectly aligned, the model that have been developed is much valid because it is showing much close resemblance to the actual template code. And the next step is after model generation, we need to refine the model and we need to validate this model. The idealization of bond geometry and removal of unfavorable non-bonded contacts can be performed by energy minimization with force fields such as CHAM, amber, and chromos. So after aligning the protein onto the template protein, we need to minimize the energy or the force fields, right? After getting a proper model, we need to see that the modeled protein or homology is having minimized energy. Meaning minimized energy, it is a minimized energy in the sense it is the most stable form. We need to convert it into the most stable form by using these softwares like the CHAM, amber, or the chromos. And there might be certain deviations from the ideal stereochemistry values in terms of bond lengths as well as angles. That can also be identified by using the tools like ProCheck, Prove, and What If Ramchandran plots. These are utilized to identify if there are any deviations from the ideal stereochemical values for bond lengths as well as the angles, right? So these two softwares are helpful in order to refine this and validate this particular developed model. And the homology model validation, as said previously, we can do it by ProCheck, or we also have different plots like a RAT plot, ProSci plot, as well as Verify 3D, What Check, and What If, right? And these are some of the softwares for homology modeling, that is Modeler, Prime by Schrodinger, that is Coverage Studio, and we also have Molecular Operating Environment and Swiss Model Server. In this workshop, next Mr. Dilip will take you into how we can use these softwares, in particular the Prime by Schrodinger as well as Swiss Model Server, in order to perform this homology model. I hope everything is clear, and now I will give the screen to Dr. S. Dilip in order to move forward. Thank you. We'll have questions at the end. Video.
Yes. Good afternoon, all of you. Myself, Dr. Dilip. First of all, I thank Dr. Basha sir for wonderful introduction related to CAD. Now I will share my thoughts related to some of the software which we can employ for the CAD, that is computer aided drug discovery and development. And Sir already explained about some basic things in homology modeling concept. And here I will share some of the projects which we have done using Schrodinger as well as Swiss model. Swiss model is online web server where we can perform homology modeling, and Schrodinger is paid software. There also there is a module name as Prime, which we can employ for the generation of different homology model. This is my first project. The title of the project is Development of Homology Model for the enzyme alpha glucosidase of Saccharomyces cerevisiae, that is Baker's yeast. Here, alpha glucosidase is one of the best target or best protein for the treatment of diabetes. As we know, diabetes is one of the global burden. There are a number of drugs are there for treating diabetes, but they have their own limitations as well. And there are some of the inhibitors like acarvos, oglibos, miglitol. These are some of the alpha glucosidase inhibitors, but they have some drawbacks like different kind of side effects. In order to mitigate that side effects or in order to reduce that side effects, scientists are very much interested to develop novel molecules or new chemical entities to treat diabetes. But here there are number of targets were there, in out of which alpha glucosidase is one of the target where we can use the target to treat diabetes. If we develop some of the molecules or new chemical entities with safer like intake, if efficiently, if it inhibits alpha glucosidase, means we can get good output and we can treat diabetes. Here we can say comparative protein structure modeling was performed with the help of Prime Homology Model tool of Schrodinger suite. Prime is the module which was present in Schrodinger suite. We have used that Prime module for the development of homology model. Firstly, due to unavailability of the 3D structure, there is no crystal structure is there for alpha glucosidase enzyme. There is no crystal structure. So that the reason we have developed our own model. We have developed our own homology model for the alpha glucosidase enzyme of Saccharomyces cerevisiae. If the crystal structure were there, for suppose there are a number of targets were there for the treatment of diabetes. So many targets have their own crystal structures. That means they have their own PDB coordinates. But in case of alpha glucosidase, there is no reported crystal structures were there. Then we took the challenge to develop our own model for the modeling studies. Here, the amino acid sequence, of alpha glucosidase of Saccharomyces cerevisiae was obtained from UniProt. Here, each and every protein has their unique identity. Each and every protein has their unique identity. You can see UniProt ID. The UniProt ID of Saccharomyces cerevisiae is the UniProt ID of Saccharomyces cerevisiae is P53341. If you use that P53341, where we can get the amino acid sequence of particular alpha glucosidase enzyme. Then based on literature or other databases, we can get the UniProt ID and we can extract the extract the amino acid sequence of query protein, that is alpha glucosidase enzyme. After query which we have identified, then we need to perform blast sets. Blast sets means like a basic local alignment search tool. It is responsible for the searching of most resemble template. It will identify the most resemble template of query sequence. Query sequence means which we need to produce the homology model. Template means which we want which we are using for the for the development of homology model of alpha glucosidase. After identifying the query sequence, then we have done blast homology model, blast homology sets in order to identify the best template for the alpha glucosidase enzyme. Here you can see PDB ID of 3A, 4A. 3A, 4A, this PDB ID belongs to isomaltase of Saccharomyces cerevisiae. Then we have utilized this 3A, 4A PDB ID for the development of homology modeling. Then after developing homology model, we have used Ramchandran plots and other protein check plots for the evaluate the model 3D structure of investigating protein. And here we can see the 3A, 4A, it has resolution of 1.6 angstrom units, and it is sharing 72% sequence identity with alpha glucosidase enzyme, that is query sequence. And here you can see sequence alignment of alpha glucosidase from Saccharomyces cerevisiae and isomaltase from Saccharomyces cerevisiae. Here SPP53341 represents for query sequence. What? For query sequence, that is alpha glucosidase enzyme amino acid sequence. 3A, 4A represents PDB ID of template. What are the template which we found through blasts? That is the 3A, 4A. And here you can see the similarity between these two. Right, there are number of amino acids are similar. They have similar sequence, that is alpha glucosidase as well as isomaltase of Saccharomyces. They are sharing similarity in arrangement of amino acids. And after this sequence alignment, we have performed homology modeling. Then we have developed a new model. Here you can see the ribbon structures. It represents the new homology model for the alpha glucosidase enzyme. After developing that new homology model, we need to evaluate that model. Whatever model we have developed, is that correct or not? We need to evaluate that model. Then we have evaluated through Ramchandran plot. You can see here Ramchandran plot, that white region represents energetically low favorable, energetically unfavorable or low favorable. And here yellow region represents energetically favorable or favorable region. And here that red region represents most favorable region. If you see the dots represents number of amino acids, and the number of amino acids present in red region is very, very high. That means whatever developed model, it is having good resemblance. And the Ramchandra plot of developed model indicated that 98.1% of the amino acids residues are in the most favored region, and 1.7% of the residues are in allowed region, and only 0.2% of the residues are in disallowed region. And this way we have developed homology model, and we have published this work in Journal of Molecular Structure. Now we are going for some small demonstration by using Schrodinger software. screen. Here you can see the screen of Schrodinger. Yeah, this is the like a prime screen we are seeing here, and there are number of options for there. There are number of options like file, edit, select, workspace, script, view, and window, and other options. And here you can see task options. Here you can see in right corner, you can see tasks options where you can find the homology modeling option. Okay. Here you can find the homology model. First, we need to download the query sequence. Then there is a online web portal that is Uniprot. Then just log into Uniprot once. There you need to enter Uniprot ID of query sequence of alpha glucosidase enzyme. Then just click search. Then here you can see alpha glucosidase enzyme of organism Saccharomyces cerevisiae, that is Baker's yeast. Where you can see format. This option is for downloading faster sequence of alpha glucosidase enzyme. Go to format, and here you can see option faster canonical. Then just click that. Then you will get amino acid sequence for alpha glucosidase enzyme. You can see yeast, Baker's yeast, and its Uniprot ID and alpha glucosidase enzyme, right, of Saccharomyces cerevisiae. and this is its amino acid sequence for that particular query alpha glucosidase enzyme. Now I am downloading this FASTA sequence. I have saved that FASTA sequence. Then we need to, here you can see blast cells. Here you can see blast. The second option is blast. Then you need to click that blast. It will take few minutes for identifying best template. This blast search is for identifying templates. We know the query, but we want to know about template. Then we are, we are searching template, best template for the alpha glucosidase. Then it will take a few minutes to give the results. It is identifying its best templates. It is taking quite long time. Meanwhile, we'll know about PDB data bank. PDB data, protein data bank. PDB also known as protein data bank. Here you can see RCSB PDB, protein data bank, where all the protein crystal structures will be deposited. Right? We can here we can see number of protein crystal structures. If any scientist, if they will solve crystal structures, they should definitely deposit their crystal structures in this protein data bank. They will get prior approval and they will deposit their crystal structures. And each crystal structure has its own PDB ID or PDB coordinates. For suppose, like if you consider like topoisomerase enzyme, it has its own PDB IDs, different PDB IDs. Okay. It is taking quite long time. First we need to do like this. Then we will get the number of templates which are resembles to the query sequence. We can identify from that. Then I have done this study earlier itself. Then I know which one is best template. I can get the coordinates from here. Then there is a PDB ID called 3A4A. This 3A4A is belongs to isomaltase of Saccharomyces cerevisiae, and it has resemblance of 72% with query sequence that is alpha glucosidase. Then we need to download this PDB ID. You can see download file 3A4A, that is PDB ID, and that it is crystal structure of isomaltase from Saccharomyces cerevisiae, and it is originated from Saccharomyces cerevisiae, and it is expressed system is Escherichia coli. and its resolution is 1.60, and its resolution is 1.60. And then we have we have to download this PDB ID. Go to download option, then you will get this, then PDB format. Right here you can see PDB structure for 3A, 4A. Now I am copying this structure to the folder. That is Homology Modeling folder. Right. First task was done. That means first we took faster sequence of paraprotein, that is alpha glucosidase, as well as we have done blast homology search. Still, I guess still it is running. Right, we got the results from the blast search. Here you can see the resemblance of paraprotein with the template. There is a P53341, that is a target which we want to do for, and there are other targets also were also there. But before going for for all the final studies, we need to evaluate the best PDB IDs. Right? We have I have evaluated most of the PDB IDs from the resemblance PDB structures, but I got best results for this one. IMA1. Here you can see oligo 1,6-glucosidase IMA1 isomaltase 1. Here we can see this protein. We can take it as a template. Here you can see PDB IDs for the particular isomaltase enzyme, that is 3A, 4A. Right, here you can see that 3A, 4A, 3A, 47, 3A, 4A, 3A, Z7, 3A, X6, 3A, X1. These are different PDB IDs which were related to the isomaltase enzyme. Isomaltase enzyme. I have tried with 3A, 4A and 3A, 3A, 47 and 3A, 4A. And we got the best results for 3A, 4A. That is PDB ID of 3A, 4A. We got the best results. It has resolution around 1.60 angstrom units. And now we are using that PDB ID as a template. Then we have downloaded that PDB ID, that PDB structure from PDB protein data bank, that is 3A, 4A. We have downloaded and we have saved that structure in my homology modeling folder. Then coming to the software session, then first one we need to click homology modeling. There you will get two options like multiple sequence viewer and structure prediction wizard. Then click structure prediction wizard. Then you will get the window like this. You'll get the window like this. Then you just need to go to file, and I have already performed earlier, that's why it is coming this one. File, go to new option, then run to, click OK, and then there is a empty window. There you can get faster sequence of periprotein. Here you can see get sequence from. You can see get sequence from. Just click file. Then go to desktop. There I have saved this faster sequence that is for periprotein. Now I have imported that periprotein. Here you can see it has 584 amino acid sequence in it. You can see P53341, that is uniprot ID of that peri sequence, that is alpha glucosidase enzyme. It has 584 amino acids. Then click next. Then you will get this kind of window where you can import file of PDB ID of all isomaltase enzyme. Okay, here you can see this window is for importing template from file. This window is for importing template. You can see from file, you can import our template. (3A4A), that is a template which has more than 70% resemblance with the peri sequence. We are importing this template now. Here you can see the sequence alignment. Here you can see the sequence alignment of starting query sequence as well as template sequence. 3A4A. These amino acids belongs to template sequence, as well as amino acids of this one belongs to the query sequence of alpha glucosidase enzyme. Here you can see the similar color things represents, they have similar sequence. Like there are, there are different amino acids are also there. There are, we need to do model for that. After this, you need to click next option. Then there are different kind of options where you can cut the unwanted amino acids, or if you have any like unwanted or any heteroatoms which are there in peri sequence, which we can eliminate using this crop option. Otherwise, simply you can click next option again. Then there is a option called build model. Build model. That means it will create new model for the peri sequence. If we click this one, job was started, where you can see here it will, for completion of job, it will take quite time, but already we have performed this job like a few hours back. You can see in workspace. Yes, right now it started, but for completion of this job, it will take quite time. But already here you can see the already prepared model. After completion of the job, we'll get this kind of structure. We'll get this kind of structure. Okay. This is the structure which we have prepared earlier. You can see the homology model of alpha glucosidase. And this model, we need to check Ramachandran plots of this or the other tools which we can evaluate the prepared model. Like prepared model is good or not, we need to evaluate. Then just click task. Then we can enter like Ramachandran plot, like these softwares will take more memory. That's why. Then you will get option like Ramachandran plot. Then click that Ramachandran plot. It is taking quite time. After clicking that Ramachandran plot, you will get the plot like already we have seen in presentation. Right. In this presentation, we already have seen this is the Ramachandran plot for the alpha glucosidase enzyme, which we have prepared through homology modeling technique. Right, and here you can see most of the amino acids are energetically favorable region, that is in red region. Very few number of amino acids are out of that region. Okay, so that we can say like that our prepared model is good. Our prepared model is good. For suppose if number of amino acids are out of the range, like if they are in unfavorable region, if the number of amino acids are unfavorable region, then we need to do loop refinement. There is an option called loop refinement in Schrodinger, where we can refine the loops so that all the amino acids which are in unfavorable region comes to the favorable region. Okay, that option also will be there in Schrodinger. We can use that option to refine the loops and to refine the entire prepared model. Okay, this way we will do homology modeling for like any unknown proteins in order to get the good model. This explanation is for Schrodinger. Then we'll see this is for Schrodinger. Then we'll check with Swiss model. That is also one of the well-established online tool for the performing homology modeling. Just go to Google. Then here you need to add like Swiss model server. And you will get the first option. Just go to that. Then you can see number of options and click start modeling. and he'll get the window like this. And there are options like target sequence and project title and email and search for templates and build model. Here, target sequence is our query protein amino acid sequence, that is alpha glucosidase, which we took as a query, and we'll paste that, that sequence here. This is the query sequence. Copying the sequence and pasting in the Swiss model server. Then I'm entering like any name, project title, and the email option is optional, and you can click search for template. Here option is the search for template. Then it will take quite time for the searching best template. And Swiss model is one of the most authenticated for the generation of homology models. And we can utilize that models for the other modeling studies. After building that model, we can check with molecular docking or molecular dynamics or other other utilizations like for modeling also we can do by using that kind of models. Right here you can see the things like most favorable templates along with their PDB IDs also we got. Right here you can see 3AXH, 3AZ7 and 3AZ7, 3AJ7, 3AXH. These are PDB IDs of particular proteins, and we got around more than 50 templates we got, out of which we can see 3AXH it got first rank, and it is resembles around like similarity around 72%, and this also and this protein also belongs to isomaltase, glucosidase isomaltase. IMA represents isomaltase enzyme. That means in like normal blast like basic local alignment sets to also we got the isomalt as a best template, and in case of Swiss model predictions also we got the isomalt as a best template. And then we need to click like which one we want to use as a template. We need to click on it. Okay, then I took first one as a template. Okay, then if you want any other PDB IDs as a template, you can click on that PDB IDs, then just click build model. Then you need to go for build model. Then it will take quite time. Right, we got the results. Here you can see the generated model. This is the generated model. Okay, this model we have generated through online web portal, that is this model. And before that we have learned about prime module of Schrodinger. Then we can download this one by clicking structure assessment. By clicking structure assessment, we can download the PDB format of that. Otherwise, you just click model one and keep it as a download. And here you can see the Ramchandran plot for the generated model. We can see the Ramchandran plot for the generated model. Here, thing is that here that green color represents most favorable region. Light green color represents allowable region, and this region, like almost equals to white, it represents unallowed region. Okay, here also we can see most of the amino acids are present within the range. That means within the favorable region only they were there. That means the generated model is good. Okay. Then we can download this PDB from this portal, and we can utilize that PDB for the next steps like molecular docking, molecular dynamics, or pharmacophore modeling, and other for other steps we can utilize these models. Okay. Now we have learned about prime of Schrodinger and Swiss model of Web 2. We have learned in this session. With this, I am concluding my session. In this session, we are completely focused on CAD, like computer-aided drug design, in the development of novel drug molecules, and in that mainly focused on homology modeling. Homology modeling tool is one of the technique where we can build our own model, like in the absence of any particular crystal structures for the protein, we can develop our own models. And this technique is a well established one. And in that we have seen what are the softwares where we can utilize for the performing homology modeling, and the steps involved in the homology modeling also we have seen. And we also discussed about what are the what is the importance of homology modeling, as well as like a prime model of Schrodinger, as well as Swiss model of online vector also we have learned. If you have any audience, please, if you have any doubts related to performing homology modeling or related to CAD tools, you just keep message or you can free to contact. And in this regard, I'm concluding my session. I would like to thank Andhra Pradesh State Skill Development Corporation, all the coordinates of APSSPC, as well as Vishwanath sir, coordinator, VPC, Indian Pharmacy College, and beloved principal sir, and Basha sir for wonderful introduction. If you have any doubts, you can free to call or message.
Dear participants, any doubts? Can you ask? లేదంటే చాట్ బాక్స్ లో మీరు మెసేజ్ చేయండి. అలాగే గూగుల్ ఫామ్ ఫీడ్బ్యాక్ ఫామ్ కూడా సెండ్ చేయడం జరిగింది, మీరు ఫిల్ చేయండి. ఎనీ డౌట్స్ డియర్ పార్టిసిపేషన్స్?
If you have any questions, we are happy to answer.
సార్ వెయిట్ చేద్దాం సార్, ఒక 2 minutes
Yeah,
వెయిట్ చేద్దాం సార్.
yeah, we will do. No problem.
ఓకే సార్.
Yeah.
షూర్ అడగండి అండి, మీరు తెలుగులో కూడా క్వశ్చన్స్ అడగవచ్చు. మీరు మైక్ మ్యూట్ చేసి మాట్లాడవచ్చు. మీరు రైజ్ హ్యాండ్ చేయండి, నేను మీతో మాట్లాడే ఛాన్స్ ఉంటుంది, సార్తో మాట్లాడవచ్చు మీరు. సో మీరు రైజ్ యువర్ హ్యాండ్ చేయండి, రైజ్ యువర్ హ్యాండ్.
ఏదైనా ఒక డ్రగ్ ని మార్కెట్ లోకి రిలీజ్ చేయాలంటే తప్పనిసరిగా 13-14 years పడుతుందా సార్?
Yes, sir. It will take 13 to 15 years for from discovery phase to drug development. Around it will take 13 to 15 years, sir, for single drug molecule.
Okay, sir.
Because there are multiple steps that are involved. Because it's not just like that. We have many things to be considered, like synthetic part, testing part. Not all the compounds which we test, they have good activity. There are only certain compounds that will come out of our, say for example, you have screened 1,000 compounds, not the 1,000 compounds will have good activity. Then we need to go for, new synthesis again. Like that, this is a typical thing. But computational methods have enabled us to decrease the time comparatively. It is the advantage of computer-aided drug design.
సార్ రీసెంట్ గా కోవిడ్ వచ్చింది కదా సార్, కోవిడ్ కి వ్యాక్సిన్ కనుక్కున్నారు, ఇంత త్వరగా దీనికి మరి ఎలా చేస్తున్నారు సార్?
Actually, like one of the project we have handled, sir. where you are like using a like compound where you can utilize that compound for the treatment of COVID. Also, we wrote one paper. Then if you'll identify through CAD also, we need to test biologically, sir. Like for suppose we identified through CAD one of the molecule, it has best score and best efficiency towards the COVID proteins. But for ultimately we need to test it on biological systems, sir. That should be there.
So your point is whenever it comes to a global emergency, as it is global emergency, there will also some flexibility in the, rules that will be taken into consideration. Because as it is a global emergency. So the regulatory authorities as well as the speed of the process, because it will be specially funded for these vaccines. ఇంత ముందలంటే vaccines కి ఎవరు అంత డబ్బు పెట్టరు, కానీ as it is a global emergency, all the scientists and the governments of all the countries will come together and share their knowledge equally. ఎప్పుడైతే నాలెడ్జ్ షేరింగ్ అనేది ఓపెన్ గా ఈజీగా జరుగుతుందో ద ప్రాసెస్ విల్ యాక్సలరేట్. ఏదైనా ఫాస్ట్ గా జరగడానికి అవకాశం ఉంది. దట్ ఈస్ ద రీజన్ వై వీఆర్ ఏబుల్ టు గెట్ ఫాస్ట్ వ్యాక్సిన్స్ ఇన్ టు ద మార్కెట్. Previously, if you take one company, it will do its research on its own and it will comfortably deliver its results. But now the condition is different. COVID is totally a different scenario where everybody should work together in order to counteract that disease. That is how the governments as well as the scientists and the researchers are working together so that we can resolve all these problems as soon as possible. That is the reason why you are able to get vaccine early.
Okay.
Hope the answer is clear for you. Thank you.
Okay, thank you so much.
Yes, yes.
Dear participants, any doubts?
If you have any doubts also, you can contact later. We will be happy to discuss and get into this so that we can share among ourselves and get a good understanding of the topics. And we can also gain new knowledge from your ideas so that we can do better research. We are always open and you're always welcome with respect to these tools or any sort of doubts or any sort of collaboration.
Thank you, sir.
Much happy to do so.
Thank you, sir. Thank you, Abdul sir and Dilip sir. Thank you once again, sir. And tomorrow we'll session continue, sir. Okay,
thank you, sir. Thank you.
Thank you.
We'll continue tomorrow. Have a nice time.
Thank you, sir. Bye, bye, bye.
Thank you. Bye, bye.